Hi, thank you. I'm Brad Glasscock. I'm the head of regulatory affairs at BioMarin. It's a privilege to be here to be able to present this case study from our most recent product approval, Palynziq, and to be the first speaker after lunch. So thanks for the coveted postprandial lineup in the. So hopefully I can keep everybody awake here. If not, Frank Sesnowski, I think, has a 2:45 time slot. Maybe he can do some stand-up for us. So I'll start today with a brief introduction to BioMarin, who we are, what we do, and what products we have available. I'll follow with a case study, again focusing on the recent approval of Palynziq for PKU, and then follow that with lessons learned from this experience. And I'll close with some recommendations and observations to share with the few FDA participants that I think remained with us for the afternoon. So BioMarin is a company that's focused in the development of orphan products with a particular focus in rare genetic diseases. We were founded in 1997 and we're headquartered in Marin County, hence the name BioMarin. Our current size is over 2,700 employees worldwide. And we have clinical or commercial operations in greater than 70 countries throughout the world. We currently have 6 products approved in the US, including 3 which have been approved in the last 5 years. Vimizim, Brineura for CLN2, which you heard a little bit about this morning, and then Palynziq, which will be the focus of the case study today. So a little bit of disease background. Palynziq is approved for the treatment of adults suffering from phenylketonuria, or PKU, a rare and serious genetic disease. It's characterized by the body's inability to break down phenylalanine, or Phe, and is caused by a mutation in the gene encoding for phenylalanine hydroxylase, or PAH. If left untreated, this accumulation of Phe results in intellectual disability, psychiatric and neurological symptoms. One of the mainstays of therapy is severe dietary restriction of Phe intake and protein. However, as patients get older, it becomes increasingly more difficult to adhere to this diet. And the overall treatment goal throughout a patient's lifetime is metabolic control as measured by a reduction in phenylalanine. So this is just a picture depicting the vicious cycle associated with the disease, starting with a lack of compliance with diet, which then leads to an exacerbation of hyperphenylalanemia, which then leads to development and worsening of psychiatric and neurological symptoms, which then feed back into further difficulty in complying with this diet. Palynziq was approved in May of this year, again in adults suffering from PKU. The approval was based primarily on the effects on the primary endpoint, which was considered a surrogate endpoint, which was blood phenylalanine levels. And I've noted in these 2 bullets, bullet 2 and 3 here, just some quotations from FDA's summary basis of approval. The first coming from the office director, noting the dramatic effects in Palynziq's ability to lower Phe levels, which thus represented a major therapeutic advance in the treatment of these patients. The second quote here, the last bullet on the slide, comes from the division director, who acknowledged that in PKU, the standard of care and really the goal of treatment is metabolic control, is measured by Phe levels, and which is clearly articulated in current treatment guidelines. So I wanted to spend a couple minutes just highlighting the basis for using Phe as a surrogate endpoint that is known to predict clinical benefit. Now, a lot of times when we talk about surrogate endpoints, we talk about reasonably likely. In this case, because we received traditional or full approval, Phe was considered a higher threshold, which is a surrogate endpoint known to predict clinical benefit. And so this slide will outline the 3 key pillars of that support that we put forward to justify that position. The first is one that you hear a lot about when surrogate endpoints are mentioned, and that is, when a disease has a well-characterized pathophysiology and the mechanism of action for the product in which is trying to affect that pathophysiology is directly related to that effect, it allows for an ability to draw that connection between expectations on clinical benefit. And in the case of Palynziq, what we have, which is basically an enzyme substitution therapy, is a correction of the underlying metabolic deficiency, and thus a correction back to the normal state. And this is substantiated through compelling clinical data which we generated from our phase 3 program, showing that the dramatic reduction in Phe levels and the restoration of the missing physiological activity. The next two pillars of support come from the current treatment guidelines, as well as the growing body of literature that speaks to the linkages between uncontrolled blood Phe levels in adults and its association with executive dysfunction, depression, and a variety of behavioral and psychiatric problems. And I've already mentioned what the treatment guidelines state, and that's the first sub-bullet there. The third pillar of support for the surrogate endpoint was based on regulatory precedent set in 2007 with the approval of Kuvan in both pediatrics and adults. This program too used Phe levels as the primary basis to support that approval, and similar to Palynziq, it received full approval as well for using that measure of efficacy. And then lastly, as part of the support that we brought forward in the BLA for the use of this surrogate endpoint to support traditional approval, was the consideration of the challenges to endpoint identification and validation, which we heard a lot about earlier this morning, as well as a lack of validated disease-specific PRO tools in this space, which is all too often the case when studying rare diseases. Now, you would think with that fairly clear story that we would have a reasonably likely, easy time to convince the agency early on in the development timeline that Phe should be, at a minimum, reasonably likely to predict clinical benefit and more than likely be able to support a full approval based on the regulatory precedent. I show here the timeline for when the IND was submitted in 2007. This was the same year as the Kuvan approval. Again, for full approval based on reduction in Phe levels. And you can see it took us over 11 years to get over the approval hump for Palynziq based on essentially the same level of evidence. What I've noted in blue circles here are the formal type B meetings, but what's not depicted on this slide is the probably dozen or so type C interactions that we have with the review division, as well as various correspondence and back and forth of advice letters and information request letters, many of which focused on Phe as a measure of clinical efficacy. And this uncertainty regarding the acceptability of Phe as the primary basis for approval or primary basis of demonstrating clinical effect really lasted all the way through the development phase and well into the BLA review timeline. So you can imagine the amount of pressure that we were feeling on the industry side going into a BLA with that level of uncertainty, the hundreds of millions of dollars that went into the development and still not having clarity on what our primary endpoint was and what it would be able to support. And here in the bottom red box here, it wasn't until February of 2018 where FDA finally communicated their decision to accept Phe as a surrogate endpoint known to predict clinical benefit and therefore supportive of a traditional approval. That was 3 months before the PDUFA action date. This was after, again, at least a dozen interactions with the agency between the end of phase 2 meeting that took place in 2013 and the pre-BLA meeting that took place in December of 2016. So that leads me to the first lesson learned from the Palynziq review, and that is the importance of and need for effective FDA communication with sponsors. Early, timely decision-making and communication are critical, and transparency is a must. Like I said, having that uncertainty going into a BLA creates a tremendous amount of pressure and uncertainty as to what we'll be able to achieve during that review. And the potential impact of having an earlier decision is, manyfold, and I've noted some here. First of all, the trials could have been designed in a simpler fashion. The development program may have actually been expedited, and therefore the product could have been made available to the marketplace sooner than it was. And the question that it's more of a rhetorical question, but if fee lowering was accepted as an endpoint for PKU in 2007, why was this not acceptable for Palynziq until 3 months before the PDUFA action date? With basically the same supportive evidence, if anything, more supportive evidence was being generated in both the literature and the emergence of treatment guidelines in both the US and EU in this space. And the amount of effort that went into justifying that was just, it was a tremendous push on the side of BioMarin to get us over that hurdle. The second lesson learned is a point that I think many speakers touched on earlier this morning, and that is the importance of the patient voice in the process. What we did with Palynziq was to, in the BLA, include a number of patient testimonials, which we worked with the PKU Alliance, or the National PKU Alliance here in the US, which is some general requests out to that patient advocacy group, which was, do a survey of your group, those who have been treated with Palynziq, and have them share their stories as to the burden of their disease, the benefits they were receiving from the investigational therapy, the risks and the adverse event profile that they were experiencing, as well as their willingness to accept or not the benefit risk profile offered with Palynziq. So we included that as part of the BLA. We supplemented that during the BLA with a patient engagement meeting. This was a face-to-face meeting. Again, it was, we leveraged support from the National PKU Alliance. We were simply observers in the process and got the necessary parties together. And we brought about a dozen or so patients with us and their loved ones, as well as 2 of our principal investigators. That meeting took place, I think, Halloween last year, so October 31st. What we learned subsequent to that was that the FDA had a medical policy council meeting with FDA leadership just a couple days later in November, where the ultimate decision was made to accept fee as a primary basis of approval. And I'm convinced that this meeting was instrumental in helping the agency come to that conclusion. So the next 2 slides are my last 2 slides, and then I'll pause for questions. But some encouraging developments, some of which we've heard about this morning from the FDA speakers, give me some reassurance that we're heading in the right direction here and really taking a pragmatic, science-based approach to the regulatory flexibility that is often necessary in regulating and approving diseases or drugs in the treatment of rare disease. The first is one that Wilson Bryan mentioned. This is a draft guidance that was published in July, entitled Human Gene Therapy for Rare Diseases. The second guidance that was published also in July is entitled Slowly Progressive Low Prevalence Rare Diseases with Substrate Deposition that Results from Single Enzyme Defects. And both of these guidances speak to the ability of leveraging biomarkers or surrogate endpoints when there's a clear pathophysiology at play and the mechanism of action of the drug or investigational agent is targeting that primary pathophysiology. And then lastly, also in July, so July was a fairly busy month for the agency in the rare disease space, the publication of the surrogate endpoint table, which was a commitment the agency made under the 21st Century Cures Act, which listed all approvals, whether accelerated or full, that was based on the use of a surrogate endpoint to support clinical efficacy. So I really applaud the agency for putting that information out there for industry and sponsors and patient community alike to leverage and to be able to think about it as we're moving forward with our development programs. And on my last slide here, so the three major points of recommendations here are in bold. I think you've heard all of these from many of the speakers before. You'll probably hear about this from many speakers following me. And I think probably the most important is the need for consistency in applying the regulatory principles that are necessary when thinking about rare disease drug development. And this consistency takes the form, and there's many different forms and shapes to it. One is consistency across review disciplines. Janet mentioned some of that in her talk earlier this morning. Consistency between centers, between CDER and CBER. Some of these rare disease therapies, PKU is a great example. Palynziq approved as an enzyme substitution therapy, but there are several sponsors, including BioMarin, who are pursuing gene therapy, and so I would make the recommendation to CBER to leverage the lessons learned from both Kuvan and Palynziq approvals to ensure that we can avoid some of the pitfalls that we experienced during the Palynziq development phase. Communication is the second big bucket. Effective, timely, transparent. I think that's fairly obvious. Probably much easier said than done, but certainly worth repeating. And then lastly, the importance of the patient voice. And while we've had great success leveraging that patient perspective, late-stage development with our three recent approvals with Vimizim, Brineura for CLN2, and Palynziq for PKU, I really think that there's an opportunity to bring that voice earlier into the process to help shape some of the feedback and recommendations made during the development phase, rather than waiting until halfway through a BLA review or even later before the agency hears that for the first time. Because we're hearing it on the industry side much earlier because we're seeking out this input. We need, in the rare disease community, we need to leverage those relationships and those connections in the patient advocacy groups, and trying to bring that into the process earlier would be greatly appreciated. I know there are efforts underway. I think FDA has a pilot program they're working with NORD for listening, disease-specific listening sessions. As part of that, one additional recommendation I would have is how we can learn on the industry side what the takeaway messages are from that. They're talking to the same stakeholders we are. Sometimes we hear different things, but if that feedback is going to be applied to how they're going to regulate investigational products, I think it's incumbent on the industry or the agency to share that perspective with the industry.